You need to kill that.
Okay, hello, everyone. We're going to get going. It's my real pleasure to introduce Hatim Sabaawy, who's our AD for Associate Director for Translational Research at the University of Colorado Cancer Center. He's also Professor of Medicine in the Division of Medical Oncology, and he holds the Staff Endowed Chair for Cancer Research. So Hatim has only been here less than 6 months, 8 months or something like that. And before that, he was at the Robert Wood Johnson School of Medicine at Rutgers, where you were for quite a few years. But we recruited him here to lead the translational research within the Cancer Center. He brings with him really extensive experience in drug discovery, and particularly in glioblastoma and prostate cancer, and really has developed organoid models as a useful platform for reasonably high throughput drug discovery and for using it to understand the genomics of cancer as well. And I believe that's what he'll be talking about today. So I don't want to take up more of his time because we're already running a bit late, but let's welcome Hatim.
Thanks so much, James. I really appreciate the opportunity. And I am very excited to be here. I am actually thrilled to meet all of you today and share with you my vision as to how the translation research at the Cancer Center can proceed with the team, the outstanding team that is here, and some ideas on how we can move and provide the impact on patient care. This is the main mandate I believe I have. So overall, I am going to show you some of our models developed, and in the last part of the talk, I'll share with you some unpublished data on the drug discovery project that we are working on. It's not moving. Yeah, I'm sorry.
Is it on your, and it won't work?
It's here. Yeah, it's on the computer. It's not moving.
That's bizarre. You know what it was? You just had to click on the
I
screen.
use the arrows, fine.
Yeah, and it might even work with this now. I think it's for some reason, yeah, you just need to have clicked on the screen.
Sure.
Weird.
So this is my disclosure slide, and nothing relevant to this talk. When I decided to talk to you today, I wanted to look at the data from the ACS, the American Cancer Society, that usually comes out every year in the first week or second week of January. And the data shown on this slide is actually a reason for all of us to come to work every day. So what is the graph showing are the projected death from cancer in the US between 1991 and 2020. More than 4 million people were averted to die because of the cancer research that's done in this country. So this is a good reason for all of us to be in this field. But I would argue we have a lot more to do, a lot more work to do to improve on these statistics. It is projected in 2023 that more than 2 million new diagnoses of cancer in the US, and 600,000 patients will die. I would attribute these numbers to many different factors, but most importantly, it is clear that we do need to abandon the type of medicine that is one-size-fits-all type of medicine, particularly in the clinic, and particularly when we are translating data from the bench to the bedside. We need to do better on improving the diagnosis of these patients, We cannot just rely on anatomical diagnosis or pathological sites or features that don't really distinguish between patients. We need to improve both the diagnosis and the therapy and the follow-up and have it as a continuum, as a long journey between the patients and the research and the clinic. And that's how I see medicine will be in the future. There are many advances that happened in the last few years. And this slide summarizes the state-of-the-art cancer diagnosis as it is done in the best clinics. What's called the molecular tumor board will order genomic sequencing for these tumors. And this is usually done at the bulk sequencing that does not distinguish between the tumor cells and the microenvironment. And this leads to a population level type of genetics that we are getting. It does not address the heterogeneity that we have increasingly been seeing in these patients. So I would argue that the implementation of single-cell multiomics and patient-derived models, whether organoids or other patient-derived models, would provide the answer to improve on precision medicine and improve on the statistics of cancer survival and responses to therapy. We're going to get a lot of data from these. So it's going to be all about how do you integrate all these different data types into biomarkers. We still need to practice evidence-based medicine in the clinic. So we need to be able to translate these biomarkers from the data that we have here into trials that are used in the clinic. Ultimately, we should be able to use these signatures, the single-cell multiomics, the organoids as patient representative, to improve on the diagnosis and develop personalized therapies for these patients. Now, why is this important? I think in the last, this slide actually highlights literature in only the pandemic era, between 2018 or so and 2022. Many high impact, high profile papers were published showing that the tumors that we are treating in the clinic and studying on the bench are plastic. They have different cell states. They have different phenotypes. All the assays that we are doing are taking a snapshot at these states, and then they don't represent responses that we see in the clinic. So these are studies done with more precision, with single cell multiomic types of assays, and that's what we need to focus more on to improve on both the diagnosis and responses to therapy in the clinic. I want to highlight particularly those two studies in cell. This is probably many of you know the KPC mouse model of lung cancer, very famous and intensely used mouse model for studying tumorigenesis in non-small cell lung cancer. And as you can see from the Cell paper in 2020, this is from Tyler Jack's lab at MIT, that heterogeneity, the cell states are present in the very early stages, in the adenoma that are predisposing these mice and patients to non-small cell lung cancer, and is present at all levels of the evolution of these tumors. Their follow-up study just a few months ago, also in Cell, shows that this heterogeneity, the plasticity in the tumors, are not only genomically derived, but also derived from the tumor microenvironment. Indeed, their data actually show that the microenvironment is heterogeneous. In these monogenic, oligogenic type of mouse models that we are used on a daily basis in our research, imagine the complexity and the heterogeneity present in patient samples. And this is not lung cancer specific. This Cell paper shows that it is actually in 38 different cancer types, the largest to date to be done, and in many other tumor types. Breast cancer, glioblastoma, colon cancer, all have this intra-tumor heterogeneity driven mainly by genetics, but also by the epigenetic state. the epigenome is also evolving in these patient samples. So introduction to this work by John Mina and Drapkin, they theorized that this is the multi-cancer evolution model, where the hierarchical differentiation that happens during development happens continuously in tissue stem cells is now changing to become the model of differentiation at many different directions. Others introduced the work as a representation of what happens during embryonic development. And indeed, when we think about tissue stem cells and the differentiation process, it happens mimicking embryonic development. There are no fixed cell types, but there are gradients of cell types and progenitors and many different features within these. So overall, this is what I wanted to share with you as to what is driving me to think more about heterogeneity and what can we do about it, both at the bench and in the clinic. It is even more important when we try to translate the data to trials, because now we have much less numbers of patients to be involved in these trials. All molecular diagnosis will lead to a smaller number of patients in each of these trials. So we need to really improve on this Simon type of phase 1 studies, the focus on maximum tolerated doses, and all the things that in the clinic does not serve directing or personalizing cancer therapy. These are ideas that are put here. I would argue that the patient-derived models and the single-cell multiomics are key in implementing these new type of co-clinical trials and correlates within these trials. So what I'm going to show you is some of our co-clinical studies that were started at Rutgers when I was there. Some of the work is already published, some is ongoing. And these are two focused both on prostate cancer and glioblastoma. In the second part, I will describe our mouse models. Those are colonies that I actually brought here. They are currently at the breeding core. And I'm looking forward to working with all of you on these exciting models. I hope you would agree with the value of using these. In the final part, I'll describe our drug discovery approach, developing small molecule inhibitors of BMI1. So this is from a study we published last year. We like to describe these models based on where they started. So many of you probably have used spheres, what's called the sphere culture or the 3D culture. Many have used the slice culture, whether from patient samples or from mouse models. The organoids, as we like to define them, are cells that have multiple different cells, cell types within the same tissue. They include the microenvironment, and they can be continued for in culture for extended time. This field actually started many decades ago. There are organoid models since 1967. But I would attribute the recent advances to three main findings. First, the work from Sabaawy in Japan, the late Sabaawy, who actually was first to show that a single ES cell is able to self-organize into an organoid-like structure. The work from Hans Clevers, many of you know, show that a single LGR5 positive cell can generate a polyp without a tumor microenvironment by adding the factors that are secreted by the microenvironment to the extracellular matrix. And Calvin Kwa from Stanford showed that they can use the air-liquid interface to develop these organoids. When we started our work, we decided to look at tissue-specific conditions. We thought that with the colonic organoids and the mixes that are developed by the Clevers lab, they are specific to the colon. We wanted to see, are there conditions that could be specific both for prostate cancer, glioblastoma, or other tumor types? And we focused on the main pathways that are involved in development. We looked at the knockout mouse models of these tissues and what factors that should be used. And by mixing these different factors in different conditions, we identify certain protocols for generating these organoids from patients. So this diagram is actually showing what you would see in the actual organoid culture, which is a 6-day time lapse of a single cell derived organoid. They don't project very well, so the diagram describes the process. You take single cells in 96-well plate, and you continue to grow them. They have the ability to develop an organoid structure, usually within 2 weeks. Some tissue types might take longer than these. We reported on these conditions, for example, for liver type of organoids last year using specific growth factors in mix for long-term induction of primary hepatocytes and hepatocellular carcinomas. Others have reported on doing organoids from iPS cells, so you could still take fibroblasts from these patients, develop them into iPS line, and then generate organoid lines from these that represent these patients. We have also coming out in a month or so conditions for lung cancer derived organoids. Both can be done also from the mouse models and the tumors from patients. So overall, we've generated a number of protocols for generating patient-derived organoids. This slide shows some representation of these. We could also take, if you look at panel E, this is a readout for immunotherapy. So if you take biopsies from patients, combine them with TILs or lymphocytes from the peripheral blood, you could use the flow to actually detect the activity of these TILs or immune modulating cells to be able to do these assays later on in vivo in syngeneic mouse models, for example. So our goal was to develop this as part of the patient care. Many of you know, Epic is a software used for follow-up of patients in the clinic. And there are Epic modules that you can add the preclinical or the co-clinical data from each patient. So we can incorporate the development of these organoids on a patient-by-patient basis. You could implement responses to therapy and changes into clinical care. And this is one of the goals I am trying to accomplish here, working with a team at the hospital, Wells Smith and Melissa Handel and others. So the goal is to be able to use these patient-derived models in ongoing trials to develop responses to therapy, to develop readouts for many of the next line therapies that these patients need. I'm going to show you how this was done in prostate cancer and in brain tumors. This is the trial design in prostate cancer. This was done with my dear friend and colleague, Isaac Kim, who was the chair of urology at Rutgers, currently is a chair of urology at Yale. And Isaac and I developed a way that we can incorporate the PDO system into a prostate cancer clinical trial. The goal of the trial, this is a complicated slide, but I just want to show you that we basically take biopsies either from the prostatectomy or the metastatic sites. We take 3 different pieces, divide them between histological, pathological diagnosis, sequencing, and the organoid formation. And from these, develop patient-derived PDOs for each prostate cancer patient. The goal is to correlate the drug responses in the organoid system to the drug responses in the clinic. These are some of the features of the patients involved in this trial. As you can see, we were able to make organoids from very large number of these patients. Our success rate is about 84%, which is much higher than the other organoid systems developed by the MSK group, earlier published in Cell. These are some of the features of these organoids. So focus on panel B and C. These are RNA-seq data, show that they can be paired with the original tissue. that they are tumor-derived. So this is a feature of having AR positivity and loss of P63, the tumor suppressor in prostate cancer. So though we show that these organoids are derived from the tumorigenic cells and not the normal cells. Also that they secrete PSA, and that is the merge in panel D. So this is the biomarker used in the clinic for following up on patients. We can still use PSA secretion from these organoids to follow up on drug responses. Also, key features is having positivity for TMPRSS expression and making tumors when transplanted in the subrenal capsule in mice. We also developed a readout, a proteomic type of IHC. So we thought that the next line of therapy for these patients to be experimented is to target the PI3K signaling. So in order to do this, we selected patients that have mutations within the PI3K signaling pathway or don't, and referenced them to more than 65 patients with an IHC readout that is called the PI3K activity. The concept there is that you could see many mutations within the same pathway. Then you can design targeted therapy based on that. But how do you know that the pathway is active? How do you know that there are no other downstream or upstream mutations that modulate the activity? And that's why we wanted to do this readout. So we selected nodes within the PI3K signaling, terminally phospho S6, PTEN, and Stathmin. And based on that readout, the patients will be grouped into two different groups, either high or low PI3K activity. Then we designed targeted therapy targeting these mutations. And indeed, we could show that those that were selected high respond to PI3K targeted therapy. Those are low, don't respond as well. And this reflects the PSA biomarker profile in these patients. Also, as a readout, we wanted to do testing of next lines of therapies for these patients. Many prostate cancer patients who have mutations in the DNA damage repair pathway might be eligible for olaparib or any of the PARP inhibitors. So we developed the readouts for the activity of these second and third line of therapy in the organoid models, and then a go, no go type of decision that could be used in the clinic on the activity numerically for the responses in these patients. Now, this can be complemented with drug-specific readouts, for example, abiraterone, which is another line of therapy that could be used for these patients, require the presence of CYP17, which is a cytochrome P450 that could modulate the activity from one patient to the other. So by looking at the CYP17 status, you could actually have a more precise, a more personalized response to abiraterone in these patients, and that would modulate or change the readout for the PDO assay. Similarly, in glioblastoma, and I don't think I need to introduce glioblastoma to you, other than to say that the average survival for these patients is about 13 months. That current therapy have not changed for the last 24 years. That therapy is only chemotherapy with temozolomide and radiation therapy, and that there is an urgent need for developing assays and readouts that we can improve on treatments of glioblastoma. So we collected 12 patients enrolled in a precision medicine trial where we take the tissue, develop organoid models from these patients, develop a readout for targeted therapy, either as single therapy or combination therapy. And then we proposed in a program project that was submitted to the NCI to enroll these patients on best responses to therapy trial. This work is going to be resubmitted with the neurosurgery group here. I'm very excited that I've started a collaboration with a group at CU, and we are planning to move these studies to the clinic as well. So representation from the patient-derived organoids developed here was published in 2020 from my lab. And what's showing here is the combination of the next-gen sequencing data from 4 patients representing these organoids derived from 4 patients that subgrouped as a mesenchymal subtype of glioblastoma. We show that the transcription signature and the organoids represent these patients. There is microenvironmental markers that are represented in that in the transcription signature, but more importantly, that we could use the organoid models to develop combination therapy, which is immensely needed for glioblastoma. Very complicated genomically, epigenomically, and phenotypically type of a tumor that will not respond to a single type of therapy. I believe the only answer to brain tumors and many of these other tumor types is a multiple combination therapy to target all the vulnerabilities present in these tumors. So we developed these assays, and some of them are actually ongoing in the lab in trying to develop better targeted and combination therapy. And we do show that combinations are indeed much more effective than single therapy, no matter what the target is in these patient-derived organoids. We also incorporated in these organoid models biomarkers that could be useful in the clinic. For example, my lab is focused on BMI1, so if you look at the IHC, we could combine or do a multiplex IHC for both BMI1 and readouts for the targeted therapy, phospho-S6 as a readout of PI3K signaling, and show in the last panel that even if you lower the IC50 to 0.5, 50% of the IC50 originally shown with full dose, in the combination, you're still showing activity for the combined therapy at half of the dose rather than the single therapy. So this is promising because it would allow us to move away from the maximum tolerated dose type of approach that is used in the clinic. And these data would support further preclinical studies in vivo to test these combinations. So overall, we developed platforms to assess functional responses to precision therapies, to targeted therapies. I would argue that the PDO assay supports the translation and clinical trial correlative studies and could be included in the drug screening and immune modulating agents. We're studying these effects in vivo. Indeed, I will show you some of our data in vivo next part of the presentation. And I welcome partnership with many of you in further developing these assays. I point out we started partnership with Todd Pitt's lab and the core here on developing pancreatic PDO assay for looking at tumor progression and for looking at IPMN, which is a benign type of pancreatic disorder that we can develop PDO assays for to actually prevent the formation of pancreatic cancer. It's work in collaboration with Marco Del Chiaro at the surgery branch. So I'm very excited about this potential, and I look forward to even reproducing this with many other tumor types and with many of you at CU. Another resource that we are putting some effort here in, and I'm glad that Dr. Gao is attending here. He runs the genomic course, so in partnership with him, we are developing the multi-omic, single-cell multi-omic system. We acquired this year the CosMx system. This is a state-of-the-art single-cell multi-omics that allow you to go to a resolution of about 2 micron. So indeed, subcellular level of resolution and type of data that you can get with multiplexing both the RNA signature and antibodies up to 30 different targets on the same cell, done on the same slide from paraffin embedded tissue. So it'll be a unique resource, and I'm looking forward to offering it to all CU investigators to be able to develop their assays and work out. In this next part, I'm actually going to share with you some of our mouse models. And in thinking about how I introduce these mouse models and the concepts behind them, I couldn't find any better than a perspective from our dear colleague and the key researcher here at the Cancer Center, James DeGregory. I've been so excited to join CU, and James is actually one of the reasons. I've looked up to him for many years, and I know he has much impact on the understanding of how tumor suppression and how clonal evolution and looking at clonality within these tumors is really indeed a key feature. Could explain everything I described earlier about the heterogeneity and intra-tumor evolution. So I present here his perspective from 2011 that is proven to be true, that it is all about fitness, that these mutations, whether genomic or epigenomic, reduce the fitness and allow these tumors to compete better for nutrients, for space, and evolve and become the dominant clone. So thinking more about how we can model this in the lab. And I wanted to go back, as you would see, I like to always go back between the bench and the bedside, and also from the bedside and backwards. So we've seen a lot of heterogeneity in the clinic and patient samples. How do you model it in the lab? What is the right tissue? What is the right tumor type to use? So thinking about this, we have been involved in a large-scale mouse modeling project in the last 4 years or so. This mouse colony currently is at the breeding core here at CU. And the concept here is to combine the multiple tumor suppressors that are commonly found in patient samples. We have a number of strains. I would highlight the cell cycle regulators, CDKN2A and B, and ARF, which is upstream or a regulator of P53. Then combining this with P10, which is representing PI3K signaling, and NF1, which represents the EGFR KRAS type of models. So we have all of these targets, 8 different genotypes, fluxed under a tissue-specific promoter. Depending on the strain that you use, you could induce loss of tumor suppressor expression in any tissue you would desire. Currently, we have these from as a glioblastoma models using the ASCL and GFAP3, testing different tumor-initiating cells, as a non-small cell lung cancer using the surfactant protein CREART2. Then we added 2 additional genotypes to these models. We call them a cancer Cas9 rainbow mice. As I mentioned, they can be used for any tissue-specific expression, but more importantly, they incorporate the rainbow barcode that gives you hundreds of colors that could be detected by sequencing RT-PCR or flow. And also express Cas9. So that means when we take the tissues out of these mouse models and then induce them with tamoxifen, we're able to add additional guide RNA that is targeted to additional mutations that you might see in the patient samples. So now, on the background of the common mutations found in many patients, you can add a guide RNA targeting particular mutations you see in these patients and now develop murine type of models in an immune competent microenvironment that you can test these targeted therapies on a better background model. So of course, if you, I learned this many years ago from my mentors, you can't talk about any mouse model if you don't have a phenotype. So indeed, we tested, do we have a phenotype? And indeed, these mice develop glioblastoma. We are interested in BMI1. They do have glioblastoma with the BMI1 phenotype. But also because I'm interested in the hematopoiesis field based on my training, we tested if we could develop a model for CH or SHIP, clonal hematopoiesis. So the targets involved there are targets known to be involved in SHIP. And this is work I hope to collaborate with the Hemong group here on looking more into the phenotypes of these models. But initially, we do see that they have developed a gene dose dependent effect, and they have a lymphoma phenotype when we drive the loss of tumor suppressors with the VAV-CRE that drives to the hematopoietic stem cells. The phenotype is interestingly different between the heterozygous and homozygous mice. And I would argue the heterozygous mice have a SHIP type of phenotype, while the homozygous mice have a lymphoma phenotype with loss of senescence. If we look at the glioma mice that we developed and we take sections or full brains in testing if tamoxifen induction leads to rainbow expression, you could see it on that slide using the IBIS imaging system. When we take slices from these and make IHC, we indeed show loss of the tumor suppressors, the representation here from P10, and a glioma phenotype in these mice. So we are very excited about the potential of using these mouse models as a novel model that represent the mutations that are seen in many of these patients and allow to do more realistic experimentation in the bench to be translated to the clinic. We're currently working with these glioma models, doing the single cell assays and focusing on BMI1. And as I mentioned, I'm happy to collaborate with many of you if that you find use of these mouse models in your projects. In the last part of the talk, I'm going to spend 10 minutes giving you about introduction and bigger picture overview on the drug discovery project that we have in the lab focused on developing small molecule inhibitors for BMI1. I'm excited that Jeff and the Kieft lab and others here at CU are here, and we're initiating a collaboration to really strengthen and further these studies with them. So introduction to BMI1, many of you probably don't need this, but overall BMI1 is part of the PRC1 complex. It is highly conserved. It's found in plants, in all animal species, and in all mammals. And the phenotype by loss of BMI1 is consistent. It's a dorsal phenotype based on the loss of Hox target genes, the main targets of BMI1. But BMI1 is a transcription repressor. It has many targets, indeed about 1,500 plus, and the phenotypes is mainly related to cell cycle regulation. The assembly of BMI1 within the PRC1 complex induces that phenotype at by the activity at chromatin site and has other direct targets, as I will mention. And again, these are all focused on cell survival and cell cycle regulation, as you would expect from BMI1. So what is known about BMI1 is mostly from flies. There is more work done in mammalian systems, but overall, BMI1 as part of PRC1 works at chromatin sites, and assembly of the PRC1 complex primes these chromatin sites for inducing the trimethylation done by PRC2. And that trimethylation leads to ubiquitination of gamma H2A119, and this is what induces transcriptional repression of target genes. If you actually go to the UCSD genome and look at chromatin sites for BMI1 target genes, and shown here, apologize, a little small, but the areas in purple are these areas that have repression of PRC1 activity. And as you could see, most of the Hox target genes in adults are repressed. So the phenotype in the mouse model and in mammals overall is loss of transcriptional repression when you target BMI1. There is a whole new field now, just started the last few years by 2 papers in flies, not yet, I have to admit, not reproduced yet in mammals, that there is evidence that BMI1 is involved in transcriptional activation. And this is work we've been looking at in my lab, that there are sites within the promoters of many target genes that have what's called the PREs, the polycomb repressor elements that could be induced in involved in transcriptional activation. So this is a long-term goal for the lab, is to study targets of BMI1 and how it actually balances transcription repression and transcription activation in many different cell types. Now, why study it in cancer? The answer is obvious. It's involved in more than 34 different cancer types. See bioportal data shown here that it is a proto-oncogene, highly expressed, not just in specific types, but very large percentage of patients within each tumor type have high BMI1 expression. If you look at single cell data from each tissue type, it is involved in many of these different sites, has a nucleolar type of expression by HC and upregulated in many tumor types. What we think is happening with BMI1 is that it regulates self-renewal at the tumor initiating cell level. And I don't see this as a one type expression, but it's rather a gradient. There are many pathways and factors involved. So what we think is happening with the clinic, with the current therapies, with chemo and radiation, you modulate the proliferating cells that are away from the quiescent cells. This leads to some tumor shrinkage, the cells come back. If you add targeted therapy to that, you might have some effects on these cells. The ultimate care will be a combination of personalized therapy, immunotherapy, and these types of targeted therapies. So one of the goals is to take these BMI1 inhibitors that we developed in the lab to the clinic. And this is just an overview where the work started. So we had fluorescent reporters, zebrafish embryos as a screening for effects on embryonic stem cell development. And then currently we have 2 compounds that are orally bioavailable. They are selective, potent, and can target many different tumor types that we tested. We're trying to understand the mechanism of action. We think it is through binding the UTR. I'd actually go back to this slide. You might notice here with the constructs in the primary screen, the BMI1 coding sequence is not there. We only have the untranslated region linked to luciferase or GFP. So the initial screen identified compounds that modulate translation. We think that it is through binding to the UTR. So we have ongoing studies in collaboration with the Kane lab at University of Michigan, and new collaboration here with the Kieft lab, where we want to be able to show the direct binding of the inhibitors to the UTR. Some preliminary data, isometric titration, colorimetry, direct luciferase assays. So we identify the binding pocket for this compound. If we remove it, we see loss of activity. These are data on BMI1 total cell versus ribosomal BMI1, and also a cyclohexamide chase customarily used to study the effects on RNA and translation, and competitive assays in a cell-free type of assays. Biochemically, if you look at the effects of treating GBM spheres or organoids with these compounds, you could see loss of BMI1 expression within 6 hours. You could see hyperphosphorylation at about 12 hours, and this leads to proteasomal derived degradation that is dependent on phosphatases, but not dependent on P38 kinase upstream of BMI1. The current hypothesis is that these small molecules bind to the 5' UTR. They interfere with the translation by switching the difference or the balance between the main open reading frame and untranslated open reading frame. And this leads to BMI1 hyperphosphorylation and proteasomal degradation. This is a novel mechanism that has not shown before, to my knowledge, for any small molecule targeting 5' untranslated regions. We think this effect is also dependent on the cell state or both the genotypes and the phenotypes of these cells. So some preliminary data show that indeed these compounds causes a cell cycle arrest at a nanomolar concentration. And this leads to degradation and induction of apoptosis in these cells. But using a cell cycle sensor, this is a lentivirus we have in the lab, we developed to be used in these organoid models in life assays. So we could see between the control with most of the cells in red that reflects the G2 phase of the G1S phase of the cell cycle, with the compound treatment switching to mostly green, indicating arrest in the G2M phase of the cell cycle. These are dependent on the presence or absence of CDKN2A mutations in these organoids. So we think that this will be a good readout for distinguishing between CDKN2A loss or activity in patient-derived models. And again, the goal is to develop combinations with radiation therapy and temozolomide. And indeed, we can show in these models that the combination of BMI1 inhibitors with temozolomide or with radiation at clinically relevant doses is synergistic, which is highly encouraging to move these compounds forward. As I mentioned earlier, it's important to do your validation in vivo. So we developed a PDX model from these spheres or the organoids. We show that indeed they represent in the expression profile the signatures or the IHC that is found in the same patient from which they were developed from. And using these models now, we have assays preliminarily here and ongoing using an orthotopic glioblastoma model where we can treatment with a BMI1 inhibitor as a single dose at 10 mg per kg could lead to modulation of these tumors detected by IBIS luciferase in vivo. And note in panel F and G, the effect of these compounds on the PDXs, leading to complete elimination of these PDXs. So we are excited about the potential of these inhibitors and looking forward to the next step in moving them. In summary, we developed a platform to assess functional responses to BMI1 therapy in GBM. These PDO assays support the translation and the clinical trial correlative studies and also include pharmacological screening and could be developed for testing immune modulating agents. We're studying the effects in vivo. These small molecules work at a low nanomolar concentration. They cross the blood-brain barrier. They have a high CSF plasma ratios. These are all features required for moving any therapies to the clinic in the brain tumor field. And I welcome partnership and collaboration on using these inhibitors. As I mentioned earlier, BMI1 is highly expressed in 34 different tumor types. We can't go wrong with that. Finally, I want to thank those who did the work. Linda Liu, currently here, is in my lab. She does all the glioblastoma organoids and the combination screening, a lot of expertise in the organoid models. Kelly developed the mouse models, and Gwentek worked on the prostate cancer project. I want to thank you all for all of your attention, my collaborators, sources of funding, and I would love to take any questions you might have.
Thanks so much for an excellent talk. I think it should stimulate lots of collaborations. I'm actually naturally loud, but I'm going to still grab one of these for
Sure.
passing around. If someone does ask a question without a microphone, please repeat it for the Zoom audience.
Sure.
I'll start off with one. So this BMI1 stuff is really fascinating. And so in terms of binding to the 5' UTR, I may have missed it, but have you mapped out the sequence that's required to bind?
Yeah, we have a binding pocket, a potential binding pocket. And this is part of the collaboration we're doing with the University of Michigan and with Keith Lab here, is to validate this with NMR and with other physical binding assays. This is a pre-requirement to really claim that
because you could actually also disrupt it in the cells, like with CRISPR technology, and show that now the drug does nothing. If you really want to
actually, I've showed some of these data. Maybe I went through it pretty quickly, but when we remove the binding pocket, we don't see activity.
Okay, but was that in the cells?
Yes.
Okay, I must have missed it. Yeah. Other questions from the audience? Mercedes, why don't you
please.
just shout, Mercedes, because me getting up there is gonna take a while. Yeah.
The organoid system is really interesting, and that is going to help a lot in terms of the research. But from the point of view of an immunologist, my question is how much you can reproduce the immunology, immune response within the organoid system, because that's has been always my concern is, yeah, you say, you have some immune cells also there, but which immune cells remain alive in the organoids, and how the cytokines, so all the different factors that you are using to keep the culture, what is the effect of this in the immune cells in the organoids?
Yeah, so the question is about the immune cells in the organoids and whether the conditions can reproduce the interactions of these immune cells with other cell types. And it's a great question, and it's actually an important question for the whole field. So my take on this are two ways. Number one, I need to work with immunologists here to prove it. So I really very much welcome a collaboration to do this. I've started some discussion with immunology interested groups. There are ways to maintain the immune cells within the organoid culture. I will point you to a paper from Calvin Qua's lab. First author is Neal, N-E-A-L, in Cell in 2018, and they were able to maintain the organoids and the immune cells within the culture by mixing the conditions for both of these factors. It really all comes down to the question that you have. The organoid is a great model, but what is the research question? Is the research question related, or the clinical question per se? Is it related to the interaction between these two different cell types? You have to think about the organoid model as a reductionist approach and use a co-culture assay. That is, you incubate the different cell types and do a readout between them. If you want it in the context of an immune system, this is ideally to be done in the syngeneic mouse model. You take a murine tumor, you transplant it in a competent immune system and ask the questions by flow, by taking. So I believe that two things. Models are models. They're
Mhm.
not perfect. And we can't expect them from them more than what they can give. Second, that the question should be designed to, the research question should be designed not based on model features, but based on what is the best model that could answer that question. So if you look at all of my previous record, we've never really published a paper that has one model type.
Yeah.
You
I agree.
combine the mouse models with the human models with the in vitro culture. The value of the organoid indeed is screening. It is that you are
I
able
agree.
to do a lot of things in a short time at small cost.
Yep.
Once you find your screening target, you take that in vivo, validate it, and expand on it in the most appropriate
Yep.
system. Does this answer your question?
Yep, definitely. Thank
Thank
you.
What's that? Oh, there's a question
From
online.
Zoom.
Could you read it? Because I don't have my glasses.
Yeah, of course. Yes, just someone on the Zoom asked, which domain of the BMI1 is targeted?
I'm sorry, I didn't hear.
Which domain of BMI1 is targeted?
Yeah, that's a really good, great question. So we actually developed a CRISPR, BMI1 CRISPR to many of the different targets. And it turns out many of the different domains, it turns out that the most important domain is the one that is the binding domain to ring 1B. So it's the ring domain to shortly answer the question. But I don't think it is that simple. So one of the projects in the lab is to actually figure out the effects of different mutations within the different domains and where BMI1 activity lies exactly. My speculation is that every domain is involved in different function of the many functions that BMI1 has.
Great. Were there any others? Okay. Any other questions from the audience? I know we're behind time. Sorry about the Zoom mix-up. Great. Let's thank Dr. Sabaawy for a great talk.
Thank you so much, all. Really appreciate you being here.